Nerve and Muscle Normal Histology. Myself Dr. Dipali. In nerve normal histology, we can see neuron, myelin sheath, peripheral nerves, small peripheral nerves, synapse, motor end plate, spinal ganglion and sensory receptors. The nervous system provides rapid and precise communication between different parts of the body via the action of specialized nerve called neurons. The nervous system is divided into two parts, central nervous system and peripheral nervous system. The central nervous system contains brain and spinal cord, while peripheral nervous system contains nerves and ganglia. As central nervous system is discussed in previous PG activity, so we will focus towards only peripheral nervous system. Now, neuron. A neuron is a specialized cell transmitting nerve impulses towards, otherwise called as a nerve cells. It is different from other cells as it has axons and dendrites and it does not have a centrosome, so it cannot undergo division. The nerve is a bundle of axons running in parallel in peripheral nervous system. As such, nerves do not contain neural cell bodies. Now we will see the parts of the neuron. It contains cell body, or it's also known as perikaryon, which contain a nucleus which regulates the functioning of the neuron. It has numerous dendrites and single axon. It contains nasal cell bodies in the cytoplasm. Axon carries impulses away from the cell body, while dendrites carries impulses toward the cell body. Now in this diagram, we can see this is the diagram of neuron. This is the cell body. In the cell body, we can see the nasal body, nucleus. These are the dendrites, and this is the axon. This part is known as axon hillock, where the nasal bodies are absent. In the histological diagram, we can see the dendrites, axon. These are the nasal substance and the surrounding supporting cells. Now neuroglial cells. Neuroglial cells are the supporting cells which provide insulation to the neuron. They are divided into two types depending upon where they lie. The neuroglial cells present in the central nervous system are ependymal cells, astrocytes, oligodendrocytes and microglia. The neuroglial cells present in peripheral nervous system are Schwann cells and satellite cells. The myelin sheath. Myelin sheath is a whitish, fatty, segmented sheath around most long axons. The function of myelin sheath is protection of the axon. It electrically insulates the fiber from one another. It increases the speed of impulse transmission. So in this diagram, we can see the Schwann cells are covering to the axon, forming the myelin sheath. Between two Schwann cells, the gap is known as node of Renvier. These are the satellite cells. The satellite, literal meaning of satellite is the something which lies around something else. So the satellites cover the peripheral ganglion of the nerve cell body in the peripheral nervous system. So we will see the difference between non-myelinated nerve fibers and myelinated nerve fibers. Non-myelinated nerve fibers do not possess myelin sheath, while myelinated nerve fiber possesses the myelin sheath. In non-myelinated nerve fibers, Schwann cells do not show wrapping around the axons, while in myelinated nerve fiber, Schwann cell shows wrapping around the axon. Non-myelinated nerve fiber shows only one sheath in the axis of the cylinder, and in myelinated nerve fiber, it shows two sheaths around the axis of the cylinder. Non-myelinated nerve fibers do not possess nodes and internodes, while myelinated nerve fiber shows nodes, that is node of Renvier, and the internodes. Non-myelinated nerve fibers commonly present in autonomic nervous system, while myelinated nerve fibers are present in sensory neuron and some motor neurons also. So in this diagram, we can see the Schwann cell wrap around the axon, forming a myelin which pushes the cytoplasm and nucleus to the periphery. The outer sheath of Schwann cell forms the neurilemmal sheath, which is important in regeneration and repair of the axon. So it is absent in the central nervous system. In non-myelinated axon, there is a single Schwann cell cover many axons. So it do not form a myelin sheath and neurilemmal sheath, so it cannot regenerate. So in this diagram, this is the single axon which is covered by the Schwann cells and form the myelin sheath. The gap between these two Schwann cell is the node of Ranvier. This is present in the peripheral nervous system. In the longitudinal section of the myelinated axon, we can see the nerves, and we will clearly appreciate the node of Ranvier in this diagram. These are the long, slender, elongated and pointed nucleus of the Schwann cell. Now we will move toward the actual peripheral nerve. Peripheral nerve are anatomical structure which contain afferent nerve fibers which transmit the impulse from periphery to the central nervous system, while the efferent nerve fibers transmit the impulse from the central nervous system to the periphery. So in this diagram we can see this is the axon which is surrounded by the myelin. This is covered by endoneurium, and these are the many axons together form a fascicle which is covered by the perineurium, and the fascicles together form a nerve trunk which is covered by the epineurium. So in the transverse section of this histological diagram, we can see the connective tissue which is called which covered the nerve is called as epineurium, which send the septa inside the nerve, form the fascicle which is covered by the perineurium, which is also extension of the epineurium. In the fascicles, we can see the nerve which is covered by the epineurium. These are the blood vessels. So in the cross section of the peripheral nerve, this is the axon. The axon is covered by the endoneurium, and this is the fascicle which is covered by the perineurium. This is the another transverse section of peripheral nerve. As we know, we cannot clearly differentiate the myelin sheath in H and E stain slide. So we can do the resin toluidine blue stain, where we can clearly appreciate the blue colored myelin sheath in this diagram. This is the high power view of same diagram, where we can appreciate the myelin sheath, myelinated axon and non-myelinated axon. In the longitudinal section of the peripheral nerve, we can see this is the perineurium. These are the long wavy nerves with the Schwann cell nucleus. Now we will move toward the small peripheral nerve. In this diagram, we can see the fascicle of nerve in the dermis of the skin. Here we can see the wavy nerve fibers along with the wavy Schwann cell nucleus. It is another example of the small peripheral nerve in the submucosa of the large bowel. Now we will discuss the synapse. The synapse is the region where the terminal come close to one another and transmit the impulse. It is a junction that mediate information transfer from one neuron to the another and one neuron to an effector cell. In this diagram, this is the axon covered by the myelin sheath. This is the presynaptic membrane. And this is the postsynaptic membrane. In between there is a synaptic cleft. There are two types of synaptic neurons. Presynaptic neurons which conduct impulse toward the synapse, and postsynaptic neuron which conduct the impulse away from the synapse. After that, the motor end plate. Motor end plate has similar structure as that of synapse, but it has a common effector cell, that is the muscle. One motor neuron innervate few to more than 1000 of muscle fibers depending on the precision of the movement of the muscle. So after that, spinal ganglion. Ganglion are the discrete aggregation of the neuron cell bodies outside the central nervous system. The spinal ganglions are located in the posterior nerve root of the spinal cord. In this diagram, we can see these are basophilic rounded cells are the ganglions, and this is eosinophilic wavy fibers are the nerve fibers. In the high power view, we can see abundant eosinophilic granulated cytoplasm, in which we can see the rounded nucleus with prominent nucleoli. It is surrounded by the satellite cells. Now the sympathetic ganglion. They are adjacent to the vertebral column. The nucleus is eccentrically located. The cytoplasm is also is granular, in which we can see the brown stained lipofuscin granules, and the satellite cells are scattered around the ganglion. Now in this diagram we can see this is the example of the parasympathetic ganglion. which is gastrointestinal tract ganglion, which is recognized by large nuclei with dispersed chromatin, prominent nucleoli, and with extensively basophilic cytoplasm. Now we will move towards the sensory receptors. Sensory receptors convert stimuli from environment into the afferent nerve impulses. The impulse pass into the central nervous system where they initiate appropriate voluntary or involuntary response. Now this is neuromuscular spindle. This is the complex encapsulated structure which monitor the tension in the muscle. In this diagram we can appreciate this is the capsule and these are the muscle fibers. This is the histological diagram where this is the capsule and these are the muscle fibers. This is the receptor for the tendon reflex. After that, Meissner corpuscles. These are encapsulated body in the papillary dermis. These are fast adapting and discriminatory touch and vibration receptors. In this diagram, these are the Meissner corpuscles. This is the Meissner corpuscles, while these are the sensory nerves. After that, Merkel cells. Merkel cells are present in the basal epidermis and adjacent nerve endings. These are slow adapting discriminatory touch and pressure receptors. So these are the Merkel cells and these are the myelinated nerve fibers. Now the free nerve endings. In this diagram we can clearly appreciate the free nerve endings which are the common sensory receptor for touch, pain, pressure and others. This is the Pacinian corpuscle. This is encapsulated body in the deep skin which is fast adapting for the rapid vibrations. Now we will move towards the muscle normal histology. As muscle is a connective tissue, we know it contains fibers and cytoplasm. But what is special in the muscle? The muscle is a structure of any structure of a tissue is depend upon its function. So the function of muscle fibers are the contraction for the locomotion and skeletal movement, contraction for propulsion, and the contraction for pressure regulation. So the morphological classification of muscle is striated and non-striated muscles. The functional classification is voluntary and involuntary. On the basis of the morphological and functional classification, muscle is classified into three types: skeletal muscles which are striated and voluntary, cardiac muscles which are striated and involuntary, while the smooth muscles which are non-striated and involuntary. Now we will see the skeletal muscle in brief. The large skeletal muscle is a large elongated multinucleated cells, maybe up to 1 meter in length. It has a multiple peripherally situated nuclei. The muscle fiber is lined by the membrane called sarcolemma, which is filled by the cytoplasm known as a sarcoplasm. They are voluntary muscles, so they are innervated by the alpha motor neurons. They are arranged in the fascicles with endomysium, perimysium and epimysium. The cross striations are due to the organization of the myofibrils. The mechanism of contraction is the sliding filament mechanism. So in this diagram we can see these are the individual muscle fibers which are covered by the endomysium. These muscle fibers together form a fascicle which is covered by the perimysium, and the whole muscle is covered by the epimysium. Now this is the transverse section of the skeletal muscle where we can appreciate the peripherally situated nucleus. This is the single muscle fiber which is covered by the epimysium. This is the another cross section, a transverse section of the skeletal muscle where we can appreciate the perimysium which covered the fascicle. Here this is also a single muscle fiber which is covered by endomysium. These are small blood vessels and capillaries. This is the longitudinal section of the skeletal muscle. These are the peripherally situated nucleus. Here we can see the striations. These striations are represent the overlapping of the actin and myosin microfilaments within the sarcomere. Now we will see the myofilaments, how they are arranged in the myofibrils. This zigzag line is known as the Z line, and these fibers which are either side of the Z line are the actin filaments. These thick filaments are the myosin filaments. In the middle we can see the M line. So we now see the zones. The I band comprises the actin filament which are not overlapped by myosin. A band is comprises of the myosin and the fibers which are overlapped by actin and myosin. H band is comprises of the myosin which is not overlapped by actin. Now we will see the cardiac muscles. Cardiac muscles are elongated branching cells joined by the intercalated disc. It has a single central nucleus. They are spontaneously contractile, so they are innervated by the autonomic nervous system. They form the functional syncytium. After that, the cross striations are due to the organization of the myofibrils, and the mechanism of contraction is sliding filament mechanism. Now, in this diagram, we can see this myocyte, which is of branching and single central whole nucleus, and these are the intercalated discs. In the transverse section, we can appreciate the centrally located elongated nucleus. In this longitudinal section, we can appreciate the nucleus with eosinophilic cytoplasm. These are branching myocytes. In this diagram, we can see these transverse striations called as intercalated discs. These are the specialized cell junction which join the individual cells, and in this diagram we can see this red brownish pigment which is the lysophiocin pigment. After that, we will move toward the smooth muscles. These are the small cells which are fusiform in shape. There is a single central nucleus in these muscles. It is also innervated by the autonomic nervous system. There are no striations as the myofibril meshwork anchored to the dense bodies. Cells shorten and broaden on contractions. In the transverse section, we can see the spindle-shaped cells which are having the plum-shaped nucleus with centrally located nuclei within the cytoplasm. In the longitudinal section of the smooth muscle, we can see the elongated nuclei with blunted end. The cytoplasm is fibrillar and the eosinophil. The smooth muscle myofibril composed of thin myofilament of actin and myosin. The thicker myosin filament do not line up to produce the banded or striped look. Hence, smooth muscle contain no visible striation and no striated muscle tissue. In the mesenteric stem smooth muscle, we can clearly appreciate the green color stem supporting collagenous tissue and the red muscle fibers. Smooth muscle contraction is due to there is contractile proteins are inserted in the cell membrane as a focal densities and focal adhesion densities. So when the smooth muscle is relaxed, it looks like elongated, and when it is contracted, it forms a globular shape. Now we will discuss. Now we will revise the difference between skeletal muscle and cardiac and smooth muscle. The skeletal muscles are large, elongated, multinucleated cells, maybe up to 1 meter in length. The cardiac muscles are elongated branching cells which are joined by intercalated discs. Smooth muscles are small cells and fusiform in shape. The skeletal muscle has multiple peripherally situated nucleus, while cardiac muscle and smooth muscles have a single central nucleus. Skeletal muscle and cardiac muscles are showing the cross striations due to the organization of myofibrils, while smooth muscle shows no striation. These are my references.